yeah, I joined the center in December of 2024. And ever since, the learning curve has been steep but wonderful. I've learned a lot, met a ton of people. It's been a really fantastic partnership for our lab. So today I want to tell you a short story about some of the work that we do in my lab for context, and then expand on some of the projects that we're kind of growing into as a result of the partnership with CCAS. Okay, so with that, I wanted to start by thanking the people that did this work. The students bolded right here are the students whose work I'm presenting on today. But I wanted to give a shout out to the students that actually were funded through CCAS. So Aidan Durant came as a partnership with Whitman College as a visiting student in my lab this summer. Gabby Williams also came through a partnership of people that I met through the Center of California State University, Fullerton, as well as my student Sofia Gallar, which was an Agility Fund student, and my student Maddie, who was a SURF student this summer. So lots of fantastic opportunities for my students as a result of this partnership. Also, my list of collaborators, which are the people that make this work possible, keeps growing. And that's also something that has been awesome as a result of the partnership that we have here. So thank you to all of them. And of course, thank you to the agencies for the funding and the work that makes it and the money that makes this work possible. So to give you a little bit of context, because we're kind of a little bit of an odd person in the team here of what we do synthetic chemistry, or we do synthetic inorganic chemistry. We do a lot of ligand design in the context of protein biomimicry. So the idea here is that we're not protein biochemists, but we take advantage of the vast amount of information that exists within proteins, and we leverage that to try to understand the functioning and mechanisms of the active sites of these metalloenzymes, or these proteins called metalloenzymes. So those are enzymes that code for a metal cofactor in the active site. So you have a metal in the center surrounded by amino acid residues, and then that gives you specific properties that promote catalysis. So in our lab, what we want to do is then take that information and try to build synthetic catalysts that not only mimic the structure of the active site of the enzyme, but also its function. And one of the roles that these kinds of model systems can actually play is to allow us to carefully understand the mechanism of how these enzymes operate, specifically by basically creating poorer catalysts than the enzymes themselves, slowing down the chemistry and allowing us to see and observe reactive intermediates that are relevant to the chemistry. So that's kind of what we do, and we focus specifically on first row transition metals. So the two themes in my lab, one of them focuses on this idea of moonlighting functions of metalloenzymes, which are also known as aberrant functions. So we as chemists know that molecules are molecules, and they're going to do whatever it takes for them to do the reaction based on thermodynamics and principles like that. However, in the wrong context, in enzymatic systems, or when you have specific deformations, the molecules that we've ascribed a particular function to begin to act in ways that are not necessarily beneficial for the cell. And in the context of this work, our interest is in trying to understand how some of these aberrant functions are connected to both cancer development and neurodegenerative diseases. And another sort of branch of the work, and we're trying to expand further on this, and I'll tell you about this later, is kind of thinking about how we know these compounds are functional, how we take information from the enzymes, and now trying to leverage this structural information and catalysts that we've been designing to try to do some interesting oxidation chemistry, specifically in the context of the decontamination of environmental pollutants. So the short story I want to tell you about, so lots of skipping around, but kind of give you the highlights of this project that deals with this unique dioxygenase. So why it's unique is because most metalloenzymes, when you actually do substitution of the metal center on the active site, you get reduction of activity. In a lot of the cases, you get some increase of activity in very few situations, but most of the time you either get reduction or complete disappearance of activity. However, in the case of this enzyme that we're interested in, called acid reductant dioxygenase, you substitute only one atom, in this case, one metal center, and then you get completely different regioselectivity, which is a very much a unique function that we see in this enzyme. And that kind of caught our attention when we started this work. So the enzyme ARD is involved in this methionine recycling pathway. So this essential amino acid for our body kind of has to be atom economized, and we want to recycle parts of it to do different things with it. Specifically, and the most important sort of molecules in the cycle are S-adenosylmethionine and methylthioadenosine, both relevant to the growth of cells in the case of animals, and to the proliferation and ripening of fruit in terms of plants. So you can imagine that the cycle is tightly regulated. And in the penultimate step of the cycle is where we find the enzyme of our interest and the substrate acid reductase. So when the enzyme is actually bound to iron, in the presence of O2, you get the regioselective carbon-carbon cleaving of the carbon-1, carbon-2 bond of the substrate to give you KMBT, and then producing the continuum of the cycle and formate. But in the case when you substitute the metal, as I was mentioning earlier, if you put nickel in bacterial systems, manganese and cobalt in mammalian systems, you get now the breaking and the carbon-carbon oxidative cleavage of carbons 1, 2, and 3. Only by substituting the metal ion. It's known that the structural active side of all of these metals actually looks fairly similar or almost identical, with small differences based on the metal ion itself, like you would expect. But other than that, the structure of the active side looks similar. One interesting connection to this particular off pathway, which has no known function, is the production of carbon monoxide. It turns out that this particular opportunity here of CO production would be only the second endogenous source of CO in the cell. And the problem with that is that CO is actually an anti-apoptotic signaling molecule, which has been hypothesized to be connected to some of this distinct connection between ARD's aberrant function, or this off pathway function, and cancer development and disease development, specifically the case of ovarian cancer and then hepatitis C. So what we're trying to do fundamentally is take a look at building models of these active sites and answer some of the questions that remain. There's very little structural information regarding how the metal actually binds to the substrate. There's little information in terms of the intermediates that ensue in terms of the process of oxidation, both for the on and the off pathway. There's questions about why did nature necessarily allow for this sort of substitution of metals, all three of them who are very much redox active metals known in first row transition metals, and one that is not, and whether that is actually driving the redox selectivity. The hypothesis currently suggests that that's not the case, that the metal is only acting as a Lewis acid, but lots of questions remain regarding these kinds of mechanistic questions of the enzyme. So that's what we embarked to do a few years ago when we started this project back in 2015. And there were very few, or there was actually one functional model for the enzyme, albeit not a structural model of the active site. So we published some early work in 2018, but it was only until recently that my student, Kelsey Kirsch, and other students that came from my previous institution at St. Edward's University started the design of this asymmetric ligand right here that you see right here. So this is a tetracoordinate ligand, where what we wanted to do is be a lot closer in terms of the electronics and structure to the active side of ARD. So we introduced this oxygen donating moiety. We introduced some bulk to protect us from dimerization and issues like that. And then we wanted to put some versatility, ideally, because we knew that we can do substitutions at the pyridine ring and elsewhere in the ligand to give us kind of the ability to do more systematic analysis of structure and function. So what we can do is we take the ligand and then we metallate it in the presence of its corresponding triflate salt. And that's why I have this chem draw of the triflate salt capping the metal compound to ideally give us the N3O coordination environment that emulated better the active side of ARD. So that's what my student Kelsey did, and that's where we published this nickel compound, and we've now been able to synthesize the series of nickel, iron, and cobalt complexes of this specific ligand. They all have very similar structures. You see a pentacoordinate compound capped by the triflate counterion. They're all high-spin systems, so they're compounds that are interestingly reactive. The nickel and the cobalt compound, as you can see here, based on the redox potential, are compounds that are very much stable under air or under oxygen conditions for the oxidative chemistry, so they kind of can sit there as a nickel 2. However, as you would expect, the iron has a lot more of an oxidative capacity and is very reactive to O2. And this is very much analogous what is observed with the enzyme, where the cobalt and the nickel are proposed to be less reactive towards O2, at least directly, and then the iron system is much more reactive. So we can characterize these compounds. We do a lot of the structural characterization, but what we're really interested in ultimately is seeing if we can do some of this biomimetic chemistry. So at the very top here, what you can see is the substrate of the enzyme, but this is a very difficult molecule to synthesize, incredibly air sensitive. So we work with sort of substrate surrogates, something that allows us to study a little bit more normally and under more milder conditions the mechanism of action. So again, our goal is to break and oxidatively cleave the carbons and the substrate right here that are pointed here, the red squiggly lines, in the presence of the mild oxidant O2. So I mentioned to you earlier that the nickel compound is actually non-reactive to O2 under standard conditions, so the nickel 2 itself. However, when we add the substrate to the compound in the presence of base, in this case, this is the substrate benzoin, we actually immediately activate some O2 chemistry on the compound. So what we've done basically is by adding additional electron density to the nickel, we basically shifted the redox potential of the nickel center enough to then turn on the O2 chemistry. So what we see here is just an example of a UV visible spectrum where we can immediately see the growth of these bands that are very much corresponding and assigned to now a nickel 3 type species. And in the process of this reaction, we see at least 2 different intermediates that we've actually seen in the reaction. We can monitor this reaction by NMR. where we can see the reactivity of the initial nickel compound in the presence of the substrate, and then over the course of time, we see the production of one of the carbon cleaving products for benzoin, which is benzoic acid. We also reported that this reaction can happen catalytically. So we get about 16 turnovers before we think catalyst poisoning is happening for the case of 2-HAP, 2-hydroxyacetophenone, and a little bit more limited amount of turnovers here in the case of benzoin, but it also emulates a little bit of the activity that we see enzymatically. So that's exciting, and we did some DFT calculations to try to understand what's happening in the mechanism, and what we can sort of propose two different pathways for this oxidation, one that's outer sphere where the nickel gets oxidized by oxygen in formation with superoxide that can then go through a hydrogen atom abstraction at here at this position and then do the chemistry. We have calculated that we think that the more likely pathway for reactivity is happening through the inner sphere activation of the oxygen directly by the nickel. So formation of this nickel-3 superoxo compound that then moves on to make this peroxo with the oxidation of the substrate. The peroxo then gives us the dioxetane, which leads to the carbon-carbon bond cleaving that we expected and the products that we see. And then we know that it's energetically favored for us to go back to binding the substrate again and then continue the catalytic cycle as we've observed experimentally. But this is an actually exciting product because there's actually very little precedent for coordination compounds that are capable of directly activating O2. Most of the compounds, and there's actually very few of them that can do this chemistry, come from very strongly donating organometallic compounds, whereas in coordination compounds, this reactivity has not been observed. We believe this to be a high-valent nickel-3 species activating superoxide, and we don't see any signs of redox activity at the ligand center, at least computationally and experimentally, actually, to the cyclic voltammogram, to suggest that we're going to some sort of alternative pathway. So that's actually quite exciting for us. I wanted to give a shout out to my student Jintao Jia. He's actually been diligently working on trying to figure out the project I gave him is like, let's make this very difficult to make nickel-3 compound. And he said, sure, because he's very courageous that way. He's actually looking into going into his PhD as soon as he graduates here from American. And he's been doing some beautiful work. And now he's been able to consistently show that we can make in a stable form this nickel-3 compound, a stable nickel-3 compound. This is not the superoxide I mentioned, the intermediate. This is the parent compound. And we know based on some spectroscopic studies and reactivity studies that Jintao has shown that we can actually make the nickel-3, and we're at a good place because now that's a starting point for us to launch more mechanistic studies in trying to understand the nickel-3 chemistry. So great work on his side. In terms of implications of our work, this is sort of a quick summary. We've expanded greatly the availability of models for ARD. The cobalt system actually will be the first example of a cobalt off-pathway ARD model compound. The direct O2 activation is rare, as I mentioned to you, and it actually begs a lot of questions in terms of what actually about our ligand environment made it happening. And this has kind of promoted some projects that I'll tell you about in a second. And we think that this accessibility of the high-valent nickel-3 might actually be connected to some of these moonlighting functions that have been ascribed to the enzyme. We, of course, want to continue working on the nickel-3 compound. That's work that Jintao is continuing. But what we're doing now in this kind of branch project that's coming out is that we want to explore the catalytic oxidative chemistry of these compounds in the presence of various substrates, right? What if we are able to take this mild oxidant O2 and then do potentially regioselective or stereoselective oxidation chemistry that might be possible with this nickel compound. And of course, we've shown that this reactivity is possible as well with iron, nickel, and cobalt. So that greatly expands basically our ability of thinking about the oxidative catalyst that we've generated here. But I wanted to, of course, mention the research amplification that has occurred as a result of me joining CCAS in December. I mean, like I said, it was a really steep curve, and it continues to be a steep curve, but a really fast, fun curve in terms of the group of people that I've been able to commit both to the CCAS and the DCN. Of course, huge shout out to my colleagues in DCN for being incredibly welcoming and just amazing at everything. So thank you so much to them. So the first project that we sort of were funded for through CCAS was this Agility Fund project with my colleague Dalia Biswas at Whitman College. So she does, one of her many skills is to do computational biochemistry. And what she's trying to do with me, we're trying to work on is how to figure out how some of these intermediates or reactivity we're observing in the model systems connect with what actually might be happening at the enzymatic system, and sort of educate both directions. So to talk about the protein, talk about the model system, and see what sort of connections we can find between the two. And then also kind of launching this project where we can think about how to best design active site molecular compounds, right? So a lot of the inorganic chemistry community has been doing this intuitively to some extent, But what if we were to take detailed information about the structure of the active site and then begin to figure out what structural parameters actually make this active site work properly and give us even better models to be able to do not only biomimetic reactivity for purposes of understanding the enzyme, but also catalysis. And I mentioned to you that we had 2 students working in that in my lab this summer. The other reaction, as I mentioned to you, is this idea that we are now capable of accessing this high valent nickel state and accessing it through potentially a mild oxidant like O2. In the literature, there's been some reactivity done with MCPBA as a traditional oxidant for getting to this high valent oxygen compound. But the trick here is that the compounds that actually can do this, the organometallic compounds that are out there, and some of the coordination compounds that can do some catalytic chemistry, have vastly different ligands. So their ligand structure is really different across the board. So there's not really an understanding in terms of how we're designing the ability for this compound to access the high valent state. And that's something we want to tackle. We have some projects that are applying this idea of activating difficult bonds for degradation of polycyclic aromatic hydrocarbons, sp3 and sp2 carbon hydrogen bonds. But really what intrigues us is this idea of what kind of also oxidative chemistry we can elicit from this compound, and can we actually understand better what makes the nickel compound get to that high valent state. So for that, we've been very fortunate again to partner with people through the center. So Michael, Ryan at California State University, Fullerton, and then the Isaiah Lab at Carnegie Mellon University. We're now going to embark on this agility funded project to get things going off the ground and ask, using the tools of genetic algorithms and machine learning, to try to figure out how do we best optimize this catalyst that arrive at nickel high valent systems, because that question hasn't been answered yet. Like, people can do it. We have different compounds that can do that, but really we don't know what makes the nickel tick and what can actually make accessibility even more facile, and what kind of things we could arrive on depending on the ligand design. So as you can see, the asymmetric ligand could lend itself really nicely to some interesting chemistry as well. So we're very excited to get this project off the ground. I know that we'll have a student and Michael visit Carnegie Mellon this winter. So that's sort of the launching of that, and I appreciate them for their willingness to work and collaborate with our lab. Very briefly, wanted to tell you about this project that I think has some potential for possible collaborations. We also are interested in this other enzyme called superoxide dismutase. This enzyme is ubiquitous in all of our cells. Basically, it's capable of detoxifying this reactive oxygen species, superoxide, into hydrogen peroxide and O2. However, its aberrant functionality has been connected to the development of neurodegenerative diseases, specifically ALS, amyotrophic lateral sclerosis. Some of the hypotheses that are out there in terms of the reactivity of what the genesis of this connection between SOD and ALS come to the idea that potential protein deformations due to structural mutations are leading to active site changes that are enforcing a particular geometry on the copper center that lead or lend themselves to more aberrant reactivity. So what we did a few years ago is we built some active sites models of SOD with specific structural parameters. So there were just 2 compounds that we wanted to do, but we want to sort of much more broadly expand this understanding. Again, these were intuitively designed compounds. We think that they actually would do a good job mimicking the active site. They did indeed, but we haven't been able to systematically probe sort of this hypothesis. And what's really intriguing about this and what's exciting about that is that while it's known what the mechanism for disease development is, which is protein aggregation, what is still kind of questionable right now is the genesis of this protein aggregation system. And what is being hypothesized is that there's some reactive oxygen species that, again, are elicited as a result of structural deformations of the copper active site, things like hydroxyl radical, or these interesting long-lived radicals like carbonate radical or peroxymonocarbonate that might actually be doing some of the oxidative chemistry connected to disease. From our perspective, and hopefully the idea of expanding this perspective to projects within CCAS, we would like to take advantage of the vast amount of information available with protein mutant structures of SOD, and then begin to sort of map the idea of, like, what are the mutants that are leading to these deformations at the active site, and is it actually connected to some of the aberrant reactivity that we observed for SOD. So just a project that we're hoping to pitch out there for anybody interested, and I'll continue to sort of try to talk to colleagues about working with somebody on this. And then last but not least, this project that we started with my colleague Rachel Burks. Again, as it was mentioned earlier, she's here at American University and she's a member of the DCN, and Professor Lars Årström in Sweden. We are sort of building on work that they had done previously and developing this effective colorimetric test for using metal sensing compounds and understanding better how metal sensing compounds detect these drugs that are used kind of in field analysis and forensic chemistry. What our goal is to actually expand that vision, because there's actually a limited amount of metal sensing compounds out there, and take some of the copper compounds that we know how to make really well in our lab, and then use them to try to probe them as more sensitive cocaine detectors, cocaine and cocaine analogs. So the goal here is to not have all these false positives that arise from traditional testing, but instead try to do more intentional design of the ligand system and the ligand environment to be able to better design sensors for these types of applications. So exciting project, and it's something that we could potentially also use some partnership at the computational level to think about structure functional parameters that lead to particular function or particular properties that we want from these compounds. With that, I wanted to thank you so much for your attention. I apologize if I went over time, but I would be happy to talk offline or any other time. You can reach me at stoledo@american.edu. Thank you, everybody.